Okay. Hello, everybody. I'm really excited to have this opportunity to take part in this year's virtual conference. The next 20 minutes, I will talk about a technology and a workflow for high throughput single cell isolation and cultivation of anaerobic gut bacteria. My name is Julian. I'm a mechanical engineer by training, and during my PhD at the University of Freiburg in Germany, I worked on single cell isolation and analysis of both mammalian cells and prokaryotic cells. And this was also the time where I developed the first single cell dispensing device for label-free isolation of bacteria. Since our foundation as a university spin-off in 2014, we have been developing technology for single-cell isolation and analysis. And most of our customers use our technologies for the development of monoclonal cell lines for the production of therapeutic antibodies. However, we also provide customers with solutions for single-cell omics analysis and NGS library preparation, such as the IDOT and instrument for highly multiplexed low-volume reagent dispensing. Today, I want to mainly talk about an instrument for the isolation of microbial cells. Since last year, Cytina is part of the Selling Corporation, a young company that has grown rapidly over the last years, founded in 2016. Selling is now present in over 60 countries, and we have offices all around the world to develop new products and support our customers. Let me quickly go through the agenda of today's talk. I will start with a general introduction to single-cell dispensing, and we'll also spend a little bit of time to introduce the technology behind it. Then I will show you how this technology can be used for single-cell isolation of bacteria and other microorganisms. I will then present results from a study on gut microbiota, and I will finish my talk with a short intro to single-cell genome sequencing of individual bacteria from complex microbial samples. Here you can see a schematic that illustrates how single-cell dispensing works. And the core is a liquid dispenser that allows to generate tiny droplets. And the nozzle of the dispenser needs to be transparent such that a camera can be used to visualize, detect, and analyze cells inside the nozzle of the dispenser. Using image processing algorithms, this allows to determine whether a single cell with specific morphological and optical properties is going to be dispensed with the droplet. And finally, one needs a system to remove droplets that are empty or contain multiple cells. This can be done by removing the droplets with a vacuum suction before they can reach the target substrate like a microwave plate. Another strategy is to dispense unwanted droplets into a waste container, which requires to move back and forth between a waste position and the target microwell. While the first strategy is typically faster due to reduced axis movement, the second approach allows to recover cells that were dispensed into waste, which can be beneficial, for example, for rare cell applications. The system I will introduce today is utilizing the vacuum shutter, but in our company, we have also systems that are based on the second strategy. We and our sister companies at Selling have developed a variety of cutting-edge pico and nanoliter dispensing technologies. They all have in common that they are drop-on-demand technologies, which offer precise control of droplet volume, droplet stability, and the dispensing frequency. They differ in the droplet generation principle, which ranges from direct displacement of a liquid, like shown on the left side here, to acoustic waves, as you can see in the center, and the application of controlled pressure pulses to generate droplets. Based on these technologies, we have developed a variety of laboratory devices, as you can see here. The B.SIGHT series and the Cell-M1 are instruments for single-cell isolation and dispensing, while the I.DOT and the I.DOT Mini are instruments for low-volume reagent dispensing, for example, for cost-efficient and automated NGS library preparation. The workflow I will present today is based on the B.SIGHT single bacteria dispenser. It's a benchtop instrument that's based on the direct displacement dispensing technology I presented on the previous slide. If we zoom into the printhead here, you can see the nozzle of our disposable dispensing cartridges. We use an optical detection system similar to a bright field microscope to detect the cells in the nozzle of our dispensing cartridges. However, we can also integrate a laser for sorting bacteria based on fluorescent markers. As I mentioned before, the B.SIGHT utilizes vacuum suction to remove droplets that do not contain target cells. Let me show you in a bit more detail how this works. Here you can see a schematic of the dispenser with some cells inside the nozzle. Underneath the nozzle, we have the vacuum shutter in blue, and below you see a micro well plate. We're continuously imaging the nozzle area and using image processing, count the number of cells and analyze their size and morphological parameters. In case there is no cell in the nozzle, the pneumatic shutter remains switched on and removes a droplet before it can reach a target substrate. If there is a single cell in the nozzle that matches the properties specified by the operator, the shutter is switched off. We use a fast switching valve and the droplet can reach the target. And of course, if there's multiple cells, they are also removed via the pneumatic shutter. The B.SIGHT comes with high-resolution optics for the detection of small cells such as bacteria, archaea, or yeast. We use cartridges with a nozzle size of 20 micrometer, which allows to isolate cells between around 1 micrometer to 10 micrometer. The detection is based on bright field optics, but we can also integrate laser for fluorescent detection. And typical targets are basically any formats that match the SBS formats, such as 96 well plates, 34 well plates, or agar plates. And the throughput you can expect, just to give you a perspective here, is around 3 to 5 minutes to isolate 96 cells into a well plate. I will now show you a short video of the device in action. So I'm just starting the video. You switch the device on. In this case, it's the C side, but this is the same for all our B.SIGHT instruments. Use a new cartridge and pipe it in your cell sample. Attach it to the dispenser. We also have a system to agitate the cells inside the reservoir to prevent them from sedimentation inside the reservoir. Then insert your well plate, which is typically filled with a culture medium or a lysis buffer if you want to lyse the cells. And then you start the program, and the dispenser goes from well to well and dispenses a single cell into each well. And after a couple of minutes, you end up with a 384 to 96 well plates filled with individual cells that can be then used for further cultivation or analysis. So let's move back to the slides. All instruments use sterilized disposable cartridges to prevent cross-contamination. This is really the only part that gets into contact with the sample, and this is especially useful when working with pathogenic organisms or clinical samples. And compared to, for example, FACS sorting, a flow cytometry method, there's no risk from aerosols generated by the dispenser, since we are using drop-on-demand technology. Due to the small footprint, the instruments can be easily placed into regular flow benches and also into anaerobic workstations. And this really makes the integration into fully anaerobic workflows straightforward, as you will see later. For each single cell that is dispensed, a series of images is stored to the hard drive of the instrument to document the cell isolation process. And here you see a selection of such image series from experiments with Bacillus subtilis, Enterococcus faecalis, and E. coli. So for every single cell that's dispensed into a well, we have a series of images showing the cell approaching the nozzle, and then a final image that shows the nozzle after the cell has been dispensed. To make this a little bit more clear, I'm now showing a series of images from an experiment with Bacillus subtilis. Here you can see the cell some way above the nozzle, moves closer into the nozzle. Here it's detected by the algorithm, and the final image showing the nozzle, the empty nozzle, after the cell has been dispensed. Now, in the second part of this talk, I want to guide you through a study where one of our partners set up a high throughput cultivation workflow for the isolation of anaerobic gut bacteria. This workflow was established in the group of Professor Thomas Clavel at the University Clinic in Aachen. Here we see an overview of the entire workflow that was established. It starts with a complex microbial sample that's brought into liquid culture. The samples are then pre-cultured for up to 24 hours, or in some cases, they were processed directly. After single-cell isolation, the samples were incubated for 1 up to 7 days at 37 degrees in anaerobic environment in either liquid culture or on agar. The first round of identification is then performed with multi-mass spectrometry. And the motivation here is to isolate, culture, and study so far uncultured microbial species. Such species can be then used to either assemble synthetic microbiota. They might serve as potential candidates for the development of life therapeutics, or provide new bacterial genes and functions that can be used for other applications in biotechnology. To see whether single bacteria can be isolated and cultured under anaerobic conditions after they have been dispensed with the B.SIGHT, E. coli and Bacillus vulgatus, which is a strict anaerobe, were dispensed into 2096 well plates in liquid culture medium. The graph here shows the results from OD 600 absorbance measurements of the cultures in the plate. As you can see, a high fraction of the wells that were addressed with single cells resulted in growth, almost 80% for E. coli and 68.5% for Bacillus vulgatus. Next, 2 complex samples extracted from mice were processed using the workflow I outlined before. For each sample, cells were dispensed into both liquid medium and onto solid agar in 96 well plates. In total, 25 plates were processed, resulting in over 1,600 wells with growing colonies after 7 days of cultivation. As you can see here, more than 80% of the wells addressed with single cells from the complex samples showed growing colonies, which was quite encouraging. Out of these, approximately 1,000 samples were used for identification by multi-MS. The resulting bacterial compositions can be seen in the pie charts on this slide here. Please note that both samples came from enrichment cultures, which results in a slightly different relative abundance of individual species compared to the community present in the mice. The group is now applying this workflow for the analysis of a variety of different samples from pig, mice, and human microbiota. And for those species that do not match their multi-database, they sequence the 16S rRNA for further identification. This already resulted in several isolates of bacterial species that have not been cultured before. Here is one example. These are three previously uncultured strains that were isolated from mouse and human microbiota using this workflow. This is very recent data that has been presented earlier this year during a conference in Leipzig, Germany, by Friesel and Professor Clavel from the University of Aachen. I believe that this is super exciting since such culture-based workflows provide a great tool of finding and describing so far unknown or uncultured species in greater detail. And I guess for this audience here, it's no news that still more than 95 to 99% of all prokaryotic species are estimated to not have been cultured before. And in this so far uncultured majority, there's a huge potential for drug candidates or other bacterial functions that can be utilized in biotechnology or therapies. In the last few minutes of my talk, I want to introduce you to our platform for single-cell omics analysis and NGS library preparation. Why that? This is because single-cell sequencing of bacteria is a great tool to access the genomes on unculturable species. It basically allows you to bypass any cultivation step, which might be tedious or even impossible, and still get access to the genomes of isolated cells from complex microbial samples. Similar as before, we first isolate cells into microbial plates with the B.SIGHT, and in this case, the plates are typically PCR well plates, for example, 384 well PCR plates with conical wells, and then the IDOT low volume dispensing instrument can be used to prepare reactions for the preparation of sequencing libraries, including the distribution of barcodes in order to pool hundreds of single-cell libraries for sequencing. As I will demonstrate with an example on the next slide, this helps to significantly reduce cost for reagents and pipette tips. Here you see one example how we can dramatically cut down the cost for such analysis. Before NGS libraries can be generated, one typically needs to amplify the genomes of single cells with a whole genome amplification method. A method called multiple displacement amplification is commonly used to amplify the genomic DNA of single cells, especially those from bacteria. This is an ideal thermal amplification method using a polymerase called Phi29. And from a liquid handling's point of view, this is a very straightforward assay, which includes only 3 reagents: a lysis buffer, a neutralization buffer, and a master mix. And after the whole genome amplification, one then typically either performs PCR reactions to amplify specific genes, or continues with a NGS library preparation method to fragment the DNA, insert adapters, and clean up the libraries for sequencing. Using the B.SIGHT in combination with the IDOT, we were able to reduce the final reaction volume of this MDA reactions by a factor of 20, from previously 50 microliters to 2.5 microliters. This requires precise cell deposition into minute amounts of lysis buffer, as you can see on in the schematic here. And overall, this protocol brought down the cost for this step of the assay from $2,000 US to around $100 US. And I believe this is really an important prerequisite to make such a similar analysis available for routine use. Using this workflow, we conducted an in-house study on the analysis of different complex microbial samples, including fecal samples, a sputum sample, and a wastewater sample. And basically, after the single-cell genome amplification, we performed a PCR to sequence the 16S rRNA of individual cells. And on the right-hand side here, you can see a phylogenetic tree that was reconstructed from 16S rRNA sequences obtained from single cells isolated from a sputum sample. Such a workflow provides access to the genomes of the individual cells without the need for cultivation, as I mentioned in the introduction. And this can nicely complement the culture-based methods I described earlier in my talk. With that, I'm at the end of my talk. I hope I could provide you an introduction to single-cell dispensing and how this can be utilized to discover new, so far uncultured bacterial species. The B.SIGHT is an instrument for label-free cell isolation, which can be easily integrated into fully anaerobic and even into fully automated workflows that allow you to isolate individual cells from very complex microbial samples. Please feel free to contact us if you're interested in one of the workflows or in one of our technologies. We're happy to set up individual telecons to discuss your application, your workflows. After such a telecon, we typically offer on-site demos. Due to the travel restrictions, we can also offer online demos for all our instruments. So this is the chance to look at the instruments from basically anywhere in the world. And we're also happy to support you in setting up automated workflows for single-cell isolation, cultivation, and sequencing. Thanks a lot for your interest. I'm happy to follow up on questions via email or via individual calls.